today I'll continue with dihybrid cross. So in dihybrid cross, very quickly I'll write the first few steps which I have taken yesterday, since a revision of these steps is essential to understand further. So here, a plant with round and yellow seed was crossed with wrinkled and green seeds. Wrinkled and green seeds. So this two individuals constitute parental generation, and these are their respective genotypes, and below is the respective phenotypes. In brief, what does it mean is the alleles capital R and capital R provides a character called round seed in this plant, whereas capital Y and capital Y will result in yellow color seeds in that plant, that is Pisum style. Whereas small r and small r will provide wrinkled seeds. Which character will come in the plant due to these two alleles, small r and small r? It will result in wrinkled seeds. And small y and small y will result in a character called green color seeds. Okay. They produce gametes, and fusion of gamete will result in F1 generation offspring. This F1 generation offspring is a hybrid. It is also called as F1 hybrid, and it is heterozygous for both characters. And yesterday I also defined what is dihybrid cross. That is cross between two individuals that differ by two characters. Or you can tell dihybrid cross also has inheritance of two genes. So this is F1 generation offspring. Now, according to Mendels' third step, that is self-pollination, this F1 generation will be self-pollinated. Selfing, it is also called as selfing. Self-pollination is also called selfing. Now, each parent in this F1 generation, each parent will produce 4 gametes. And yesterday I told, like in brief, how the gametes are formed. So what can be done here is, the gametes, how they are formed is, that is individual alleles have to be written separately. That is, you have to separate the R and Y. Then further also separate R and Y. Further you get these alleles, okay, which has to be few. Further, the joining of R with Y gives a gamete RY, and joining of R and small y gives another gamete of this pair. Then second possibility, small r will combine with bigger y as well as smaller y. This will lead to additional 2 gametes. Therefore, a total of 4 gametes are produced. So ry and another is small r. Now, this parent, since it is same genotype, since it is self-pollination, it's the same genotype, and it will also give rise to 4 gametes, the same kind. That is ry, r small y, r bigger y. So these are the 4 gametes. Now, fusion of these gametes results in F2 generation offsprings. That means first F1 generation, then these will fuse and produce F2 generation in different combinations. That means this gamete will combine with this one of the offspring. Like that, the gametes will combine and then form the offsprings. So here, how is that the two generation tables be obtained? So on one side, 4 of the gametes has to be written, which corresponds to male, and on the other side, gametes has to be written. So 4 of the gametes has to be written, and a Punnett square table has to be prepared. This is the Punnett square table that has to be prepared. So it's a probability table. So all the 5, all the 4 gametes has to be written. And similarly, small r and bigger. This is the 4 gametes here. Then these 4 gametes you want to write and prepare. Stable. So this is the 4 gametes of one of the F1 parent. That is self-pollination, the male and female part. So now here, another next 4 gametes should be written here. So what is this? Ry, R small y. This is the second gamete, the third gamete, and fourth gamete. Both the gametes are similar here. Both the gametes are supposed to be similar. Now what happens? That means the fusion of gametes occur. So here will be the genotype of the offsprings. Okay, this is upon fusion of the gametes. Now you see fusion of these 2 gametes results in an offspring of this genotype. So this is the genotype of the offspring. Okay. So since you can also simultaneously write the phenotype and write it afterwards. Now similarly, this fusion of the gametes. What are the different offsprings? So how we are writing? Now you can see that capital R and small r is there, capital Y and small y. Usually dominant should be preferred. So therefore capital R, small r, capital Y, capital Y. Then capital R. And in this Punnett square, this is F2 generation. The diagonal represents heterozygote condition of the offspring. So without, like you can directly write here the diagonal of the heterozygous genotypes can be written here. Heterozygous genotype, the genotype of F1 offspring is heterozygous. So it is heterozygous at this locus also. It is heterozygous at this locus also. So it is heterozygous condition, not homozygous. So here, further, all the offsprings has to be written. So R combining and writing the offsprings. I told diagonal will be similar, so directly you can write. Diagonals will be heterozygote in all the cells, in all these cells. Diagonals will be heterozygote. So directly I am writing. Then this is these 2 gametes. Then fusion here, these 2 are right here. Then this and this you will write. Then further, also you continue to write the whole thing. So this was about the F2 generation uh, genotype of the offspring. So what is Punnett square? It is a probability table which, due to which you can find out the genotype of offsprings. Okay, genotype of offsprings can be found out. So here, once the genotype is established, even phenotype should be written. So you can see it is both are capital R, and R actually stands for round seed, which is dominating the small r, which is the wrinkled seed. Wrinkled seed. And similarly, capital Y is yellow color of the seed, and small y is for green color of the seed. Green seed. See, while writing the phenotype of this particular genotype, you should only take care when it is heterozygous condition. So one by one, R and R both are dominant, so there is no need to observe carefully. Directly you can write it. R is for round seed. So phenotype here is for round seed. Capital Y and capital Y. Now both the alleles are in a capital state. They are written in capital. If they are written in capital, it should be LOC. So therefore, phenotype here is round and yellow. Similarly, here you see round and yellow itself. Here both are, so the phenotype will be round, the round shape of the seed. Where, as you can see, heterozygous condition has come. So when heterozygous condition comes, you have to take care and find out what phenotype you have to write. Because in heterozygous condition, you should notice which is dominant and which is recessive. So here you can see that in heterozygous condition, there is a capital Y and small y, and always the dominant allele will be masking the effect of recessive allele. Understand? So therefore, capital Y is there, small y is there. For example, this capital Y and small y. So dominating will be what? The capital Y will be dominating over the small y. So whose character will you have to write there? The character of dominating allele. Okay, dominant allele character you have to write. The dominant allele character is capital Y. Now capital Y is what? Yellow color of the seed. So when it comes to heterozygous condition, that time you have to be careful in knowing the phenotype. So here what is the phenotype? Capital Y's phenotype comes here, that is yellow. Now here you can see R is in heterozygous condition. So here also, when R is in heterozygous condition, what you have to notice as to which is dominant and which is recessive. It's a heterozygous condition. So here R is dominant over, capital R is dominant over small r. So by convention, you will be writing capital R for dominant and small r for recessive. Any capital letters basically is for dominant and any small letter is for recessive. So here capital R is dominant. So what is the character of capital R? You have to say round seed. So here you have to write round. And both are capital Y, so no need of seeing directly you can write as yellow color of the seed easily. Okay. Then next is round and yellow again. The same thing will continue for all the cells which are heterozygous. So it is round and yellow. Okay. So how is that you got round and yellow? You have to see here. Now this is one character. Same alphabet means usually refers to one character. So R is then refers to the of the seed. Y is refers to the color of the seed. Now this is heterozygous. One is dominant, another is recessive. In heterozygous condition, whose character always will express means dominant character will express, whereas recessive character will not express. Dominant character already have wrote, it is for round. So therefore, see again which is dominant and which is recessive. And whose character will come here? Dominant's character will come. So capital Y's character will come, whereas small y's character is not going to come. So capital Y's character is yellow. So round and yellow will be the genotype of this diagonal. Now similarly, round and yellow here also. Round and green. Now here there is a recessive allele. It is not a heterozygous condition, it is homozygous recessive condition. And the allele that will express only in homozygous condition is the recessive allele. Okay, this allele will express in homozygous condition only. Now in this case, therefore it will be round and green. Round and green. The next will be this one. It will be round and green here again. Round and green. Here also it is going to be round and green. Then recessive, wrinkled and yellow. Here also it is wrinkled and yellow. This is wrinkled, also wrinkled and yellow. Last one is wrinkled and green. This is again round and yellow. So they both there is a round. So there are some phenotypes. So whatever I have written in black here, it is nothing but pairing of alleles. That is the combining of an allele. So it is a genotype here. These all are genotypes. Whatever is written in black is a representation of genotype of offspring, and whatever is written in green here, it is actually a representation of phenotype. And by seeing the genotype itself, one can write the phenotype also. But while writing, special care should be taken, especially when the genotype is heterozygous. Okay then, what is the ratio? Finally, coming to what are the ratios? So ratios can be two type ratios you get here in dihybrid cross. One is supposed to be phenotypic ratio. The other one is genotypic ratio. Genotypic ratio. Two ratios will be present. Phenotypic ratio as well as genotypic ratio. A phenotypic ratio. You have to see which are similar kind among in this table. For example, round and yellow is there. So how many round and yellows are supposed to be present in this table? So after seeing into this table, what you came to know is there are total 16 offsprings formed from in the F2 generation of dihybrid cross. How 16? Because 4 of the gametes from each parent can combine in these many ways. So 16 possible offsprings of the unique genotype can be not unique, but you can see 16 possible offsprings due to. Okay, however, there is a similarity. There is a similarity between them. They are not unique. Let me tell you, they are not unique. So here, phenotypic ratio, round and yellow, how many are there? There will be 9. If you just count them, there are supposed to be 9 round and yellow. So therefore, 9 of them is round and yellow there. So next, green will be 3. So therefore you get 3 here. Then round and wrinkled and yellow. Wrinkled and yellow will be 3. Okay. And the last one will be wrinkled and green. And that is supposed to be 1. So therefore this ratio, what is obtained, is called as the phenotypic ratio. What is a phenotype? The features that appear externally appearing features can be called as a phenotype. And many times, even certain internal features can act as a phenotype, like blood group. Human blood group is an example of an internal phenotype. So therefore, this is the ratio which you get in dihybrid cross. Whereas, if you go back to monohybrid cross, there it was for monohybrid cross, it was 3:1 ratio. The phenotypic ratio was 3:1, whereas genotypic ratio was 1:2:1. Got? So this was about the phenotypic ratio. Now genotypic ratio is similar genotypes you have to know here. So in the diagonal, the genotype is almost the same. Got? So almost it is exactly the same. That is heterozygous condition. So that will be 4 in number. Then, like this, after the diagonal, one more line to continue to draw here. So these two are similar genotype. So 2 are similar. So is to 2. The next, these two these two genotypes are supposed to be similar. So again, you are going to write as 2. Then there is only one kind here. Only one unique means double recessive. That is, R also is in recessive state, Y also is in recessive state. So that is 1. This also is unique type, so it is 1. R, Y, and Y. So among the 16, the unique type will be 1, 2, 3, and 4. 4 of the genotypes are unique. They are they are exist in only one number. Okay. Now this was diagonal again. On this side of the diagonal again, here these two are similar. So this is round and yellow. These are supposed to be similar. So it is 2 again. Then these two are also similar. So diagonal, then next line and like this. Then there is again 2 units. So these two are similar. So you're going to write 2. Now this one also is unique. Only 1 is there. So you're going to write 1. Followed by again this phenotype. This one also is unique. So you're going to write 1. Good. So this is what ratio we say? We say it is genotypic ratio. Whereas in monohybrid cross, this ratio is 1:2:1. Okay. So this was about the genotypic ratio. How to know whether you have written a correct genotype or not? That will be based on the total number of offsprings. The total number of offsprings obtained here will be 16. Understand? So if the total here also is 16, it is a confirmation that you have written a correct genotype. And of course, here also it should be total of 16. Then we say that what is that? That whatever phenotype ratio and whatever genotype ratio you have written is correct. So this completes regarding what is called the dihybrid cross. So, but what you can understand, what are the various concepts you can understand through dihybrid cross? So, certain points are there. That means law of independent assortment. So next I will explain about law of independent assortment. Mendels law of independent. To get the proper result, Mendel has taken a large sampling size so that there is a credibility in the results. So here, within this, you have to get well acquainted with this table so that anything that is asked from here, you should be able to answer. For example, how many genotypes are heterozygotes? Then you are supposed to tell 4. Okay. So how many genotypes consists of? Where two recessive state of seed color is there. So seed color recessive state means this is one genotype. So this is the genotype of seed color, which is green. This can be one possibility. This can be another possibility, and this can be another possibility. So these are genotype. Now, how many of these are present where double recessive is there? So, for example, this is 2. Got? And the capital or small, this is 1. Understand? And this is also 1. Now, total how much? 4. That means proportion they are going to. Suppose the question says regarding proportions. Then out of 16, 4 are in a state of what? Recessive state of both the alleles are relevant to the seed color. Okay. So like this, any of the combinations can be asked in multiple choice question. That means they can ask where both of the seed shaped allele are dominant or there is a double dominant state. Okay. All the alleles are dominant in only one genotype. That is RRYY. Here all the alleles are in dominant state. So its proportion becomes 1 out of 16. And similarly, when all alleles are recessive, its proportion becomes 1 out of 16. Understand? So therefore, that is how you should know what fraction. Okay. So what fraction is supposed to be present? That also you should know in order to answer certain multiple choice questions. So next is law of independent assortment. Mendels law of independent assortment. Mendels law of independent assortment. What does it state? What is the statement of it? So I'll just explain what is a statement and define it. So one is, you know that this, suppose this is the genotype. Where did you observe this genotype? In the F1 generation. So what does law of independence states is one pair of trait. This is one pair of trait, and this is another pair of trait. These pair of trait encode for a particular character. Okay. So here, according to law of independent assortment, the segregation, that is separation of one pair with that of other pair. Okay, to compare, there is a level of independence. What is that means? Some segregation of one pair as well as is independent of segregation of another pair. Okay, that too during formation of gametes. What when segregation occurs during anaphase 1 and anaphase 2 of meiosis. And this is this one segregation and all will indicate whenever there is gamete formation. Okay, first R is separated, Y is separated, and then finally gametes are produced like this, and gametes are represented like this. So it means that that's what is the what you call law of independent assortment. So final statement: when two pairs of characters, when two pairs of traits come together in F1 hybrid, then segregation of one pair is independent of segregation of another pair. Okay, during the formation of gametes. So that itself is a statement of independent assortment. So what does it say? For example, suppose this pen, the gametes of this heterozygous individual is represented here, individual plant is represented here. Suppose separation of R, okay, when gametes are formed, 50% of gametes will have the R, another 50% of the gametes will have small r. So 50% of the gametes will have capital R, 50% of the gametes will have small r. Okay. Now in this capital R, 50% of it will combine with capital Y. But another 50% of it will combine with small y. Okay. Similarly, small r, half of the small r will combine with capital Y. Another half of the small r will combine with small y. Thus, resulting in 4 gametes. That too in equal proportion. So gametes are resulting in equal proportion, and this can be possible when the alleles are located on a different chromosomes. Okay, so this is also a way in which you can tell independent assortment is going on when the gametes proportions are same. So if the genes were not located on the same chromosome or they were linked genes, okay, this will be why. Because R is present in one chromosome and Y is present in another chromosome. Since R and Y are located on different chromosomes, this results in independent assortment. In case Y was on the same chromosome, R and Y were on the same chromosome, not on different chromosome. In that case, the separation of R would have been also affected the separation of Y. It means that here, in case because it is located in different chromosomes, separation of R is not affected by okay. So that is how is the statement regarding the independent assortment. Do you understand? Do you understand? So what is here is since the alleles are located on different chromosomes, segregation of one is independent of segregation of. So this was about law of independent assortment. So then, what else you should know about independent assortment apart from the definition, apart from equal proportion of gametes? So law of independent assortment is exhibited by 4 points. Apart from definition, what is that? Yeah. This law of independent assortment is exhibited by the genes that are located on different chromosomes. Okay, but if the genes are located on same chromosomes, okay, then it becomes a exception of independent assortment. So linkage is exception for independent assortment. Independent assortment. And of course, definition here, as I told, segregation of one pair is independent of segregation of other pair with their one hybrid in formation of gametes. So, this was about the independent. Now, Mendel, you know the three laws. One cross he has performed is monohybrid, and the cross he has performed is the dihybrid. With the help of monohybrid, he has established two laws, and with the help of dihybrid, he has established another law. Okay. So, which are the two laws which was established by monohybrid cross, and that is called law of dominance. So, he put forth the law of dominance after performing which experiment? Monohybrid cross. This is a monohybrid cross. Then, even law of segregation can be explained by monohybrid cross. So, two laws were based on monohybrid cross. Law of segregation. Then second is dihybrid cross. There is law of independent assortment. The second experiment was dihybrid cross, and with the help of dihybrid cross, he was able to explain law of independent assortment. So, which cross helped to establish what kind of principles? One should know it. So, here, monohybrid cross, one should know the dominance as well as law of segregation. And the dihybrid cross, one will establish the law of independent assortment. Then, I'll be explaining about test cross, that is dihybrid test cross. One more point which I want to tell you in law of independent assortment is that one pair of trait will never affect the expression of another pair of trait. So here dihybrid test cross it is. This test cross is performed in order to know the genotype and phenotype of the F1 offspring. Now consider this is the F1 offspring. Okay, but we only know its phenotype. We don't know whether it is due to homozygous or heterozygous condition. So this plant, see here, for example, Mendel saw that the plant had a round seed as well as yellow seed color, round seed shape and yellow seed color in it. But thing is, he don't know whether this phenotype is due to homozygous condition or due to heterozygous condition. Because of law of dominancy, what happens? Same characters will be expressed whether in homozygous condition or heterozygous condition. In that case, this plant, whether it is due to homozygous condition or heterozygous condition, he has to find it out. Okay. For that, he performed this test cross. Understand? So what it means here is, so he's seeing the plant, okay, and he's seeing that the seed color is yellow and the seed shape is round. He can't see into what are the leaves there. So he wanted to know whether that particular plant is due to homozygous round and yellow or it is heterozygous round and yellow. That's what he wanted to test. And that's why he does these tests called test cross. This cross is to test whether this plant, which is actually unknown here, for him visually, only phenotype was known, but genotype of a plant is not known. Phenotype we know, but we don't know why the plant is there. It is due to homozygous condition or heterozygous condition. That was not known. So therefore, to find the genotype of this unknown plant, he performs this test cross. Now, what is a test cross is, consider this is the, right now I will just take the monohybrid test cross. So this is a test cross. What is, see here, when we say dominant, see back cross means when F1 generation will be crossed with any of the parent. Okay. And if this F1 generation is crossed with dominant parent, it is called as dominant back cross. And if it is crossed with the recessive parent, it is called as test cross. So he performs what crosses? Test cross in order to detect the genotype. Because back cross is useful for in order to obtain good variety of plants. That is dominant back. So here he crosses with the recessive parent itself. So you all know in the dihybrid cross, what was the cross? This is the parent which was crossed with another parent individual. And in F1 generation, this was the genotype of offspring. Now what he does is F1 offspring will cross with the recessive parent. And that itself is what is called the test cross. So this is which one? Unknown F1 or any plant can also be tested for this test cross. But it is crossed with always the recessive parent. So this becomes a recessive parent. Now again, as usual, gametes of this, the product of this cross is analyzed. What are the offsprings of this cross is analyzed? What are the offsprings? So here the gametes produced by this unknown parent will be of course the 4 kind it will be. Okay. And by this parent only one gamete is formed. Only one gamete is formed. Now fusion of gametes results in offsprings. Now here capital R, small r, capital Y and small. This is one of the offspring. Next, capital R, small y and small y. Next offspring, capital R, small r, capital Y and small y. So next, small r, small y and small y. So how many offsprings here? 4 offsprings. Now if you see the phenotype, check the phenotype of it. This is round and yellow. And this is round and green. This is wrinkled and yellow. And this is wrinkled and green. So you get a ratio here. The genotypic ratio is of course 1:1:1, and phenotypic ratio of this test process. What is important is phenotypic ratio. The test cross ratio is called. What is it called? This is called test cross ratio. All are unique type. So therefore, we'll write a ratio as 1 is to 1 is to 1 is to 1. It means dihybrid test cross is nothing but 1 is to 1 is to 1 is to 1. If this is the outcome of this cross, if 1 is to 1 is to 1 is to 1 is the outcome of this cross, it means that this unknown plant who they were trying to find out, this unknown plant is Heterozygous. And if it is not 1 is to 1 is to 1 is to 1, it means that if it deviates from this, then it will be a focus. So if you test also, you will just take this homozygous and test. You will come to no. Okay. So therefore, this is about the dihybrid test cross. Okay. So regarding this is how you have to represent a test cross. Okay. Now finally, what is the definition? Writing part. The definition of test cross is if F1 generation offspring, it is a test cross of the cross performed to know the genotype of unknown plant. A cross performed to know the genotype of the unknown plant. Unknown plant. Unknown plant here or okay. Genotype of the unknown plant. Okay. And the second point which you should know here is a ratio. Test cross ratio is 1 is to 1 is to 1. So this completes even what is called the dihybrid test cross. Next is chromosomal theory of inheritance. Before explaining the chromosomal theory of inheritance, why was Mendel's work not accepted? Will be the next part. Mendel's work was not accepted because of following reasons. Original reasons why Mendel's work was not accepted. So Mendel died in 1884. Even after his death, his work was not recognized. Okay, so people did not acknowledge his work. But now, what is the reason why his work was not accepted? First is he has published, publicity was limited. It means that his article was published in a journal, you can tell, that has a limited circulation. So there was no wide publicity. Okay. So it had very limited circulation and did not have any wide circulation. So when the circulation isn't wide, it means even many people will not know about his work. So that means here what you should know is that the publicity was not wide enough. That is the first point. which you should know in the Mendels work. Okay, so that is related more to publicity. Then here in Mendel's work, what happened is basically he has integrated two of the subjects. One is biology, another is maths. So this integrated approach was not convincing for that time's contemporary scientists. So they were not accepting it. So mixing maths with biology was not acceptable to the then contemporary scientist. So because of that also what happens is his work was not well recognized. Okay. So first was what? He has published in a journal. Okay, it's not famous. Or you can tell journal or magazine. Journal, it is not famous at all. So which has very limited circulation. Because of which very few people will read it. So when very few people will read his work also will not, many people will not come to know his work. Okay. Second was, as I told, mixing. I mean, here what he did is he mixed mathematics in the biology. So it was not accepted by the acceptable. Okay, that time people used to study more pure sciences like purely physics, or purely natural sciences, or purely mathematics. But he tried to combine mathematics with biology. That was not convincing for the time's well-known scientists, so that they can recognize his work. That was not convincing for them. Okay then. Second is this was the second point. Now third point was that what he says is there is no blending of characters. Okay, Mendel says that there is no blending of character, but already human beings and so many examples are there where there is a lot of blending of characters. Now, what is blending of characters? I'll tell. Blending of character. So he says there is no blending of character means what? For example, if there is a tall plant and there is a dwarf plant, okay, that these two characters will not mix and give some other character like intermediate height or middle height at all. It will be purely either tall or either dwarf. There cannot be any third character at all, he says. And that itself is called no blending of character. If it is round and yellow seed, the offspring will be either round and yellow. There cannot be any intermediate character like that. So, but that time already proof was there that so many in human being itself so much blending of character is there, and various other examples were there where there is a blending of character. So he couldn't explain that why that blending of character occurs. He could not explain it. So that also was not since he couldn't explain. There's also one of the reasons why his work was not accepted. So then comes fourth reason. So he was always been stressing on one point. Mendel stresses on one point, and that point is presence of factor. What is it called? Mendelian factors. So Mendelian factors is nothing but an allele here. What we're talking. So he always stresses on unit factors or the Mendelian factors. Okay. He says a plant gets a particular character due to these factors. Unit factors will be present. And these unit factors are responsible for characterization. And but he was not able to tell where these factors will be present. That means physical proof was not provided. It means that physical existence, physical proof for presence of these factors. Where it is present? Inside the chromosome or inside the cytoplasm. Where it is actually present? The factor. He couldn't provide the proof for it. As to where there is an existence of factor. So lack of physical proof. Okay. Lack of which proof? Physical proof. Lack of physical proof for existence of factor means where that factor is exactly present, he was not able to prove it. Okay. So this is again the what you call the limitations, like why, I mean not limitations, I can tell you the reasons why his work was not accepted. So first was about what? Regarding publicity of his work. So next, no extensive publicity was there. No extensive publicity. Then second, no publicity character, no extensive publicity because of published in a journal which is not famous, just limited circulation. Then second is what is mixing mathematics with biology. Third was, what is the third one? There is no blending of characters. This was objected by that time scientists. No blending of characters. The last one was no proof for physical existence of factors. Existence of factors. No proof for physical existence of. So this also completes why Mendel's work was not accepted. So finally, his work was not accepted, but three of the scientists noticed the important work that Mendel has done. So they have rediscovered his work. And those three scientists were Hugo de Vries, Schermak, and Correns. These were the three scientists in 1900. They discovered that and found out, noticed that Mendel has done some very important work. So therefore, what happened is again the work of Mendel was given importance from 1900 onwards, and they also noticed that he has done some very important work when it comes to inheritance of characters. Then further in the next class, I'll be explaining about chromosomal theory of inheritance as well as linkage in the further classes.